indoctrinated by Joe were early 90s. There were happy years in Stanford. My kids were born at the time. Uh, they no longer resemble exactly that pose. And the kind of things I was working on are very similar to things that were mentioned, but I strayed away from the right course, and I'm going to talk to you about blood. So, and so how do I get from logic to blood? So from logic, even during my PhD, we started talking about belief revision, and that led us to probability updates, and that led me to something called Bayesian network and learning of Bayesian networks and data, and somehow that reached biology. And this is an old slide that I use that initially I thought biology is a cool problem because they have real data. I'll try to stand one location. So if you think about early years of the century, we didn't have a lot of data available, and it seems like they had real data and they cared about it. And you start asking questions about the data. I don't know how many people know what ribosome is, but you start getting essentially slowly infected, and you lose your naivety about what you're doing, and you start actually getting some sense of what you're doing, and before you know it, you start telling your collaborator what to do in their lab, and then they tell you go do it yourself, and you end up like this. So since 2009, I have my own experimental lab, and we've been studying something called chromatin. So I'll give very brief description of what that is, and then we'll see how it connects with blood. So chromatin, many people probably have seen some version of this picture. When we look at chromosomes in textbook, they look like that. It's not how they really look like in real life, but it's a big complex of DNA and proteins. And if you zoom in many, many levels, you end up with those small beads that are called nucleosomes. And those are essentially small complexes of proteins that essentially pack around 150 base pairs of DNA. And this is really boring. Why should we care about the packaging of DNA? And the reason I got involved in the packaging of DNA is that I learned from friends that the packaging of DNA is actually information substrate. The packaging is not just let's pack the DNA, but on the package, in the same way in a physical packaging of, when you move your house, you can put labels on the boxes. Cells put chemical labels on those objects that reflect how they're being used right now or in the recent past inside the cells. So cells have this problem that they have a lot of DNA, but they have to divide it, which parts are active or not, depending on what type of cell we're talking about, what is its current state, and so on. So the same piece of DNA, for example, encoding a gene for albumin, which is expressed only in liver cell, will be packaged differently in the liver where it's being actually used, and from every other cells where it's essentially packed away and compressed, and please don't bother us with this gene because we are not supposed to touch it. Okay? So it's an information that is on top of the sequence that tells you how it's being used right now within the cell. And once you start talking about information content, things get slightly more interesting. What is encoded, how it is encoded, who cares about what is encoded, all kind of questions like those. And one of the things my lab did was using methods that others developed and we improved, map many of those, each one of those names is the name of one of those flags, map those flags along the genome, and we could look at how genes that are active look like, genes that are repressed look like, and we can see differences. So a gene that is on and a gene that is off, they are very different in terms of these modifications. And again, this squiggly line just says how much the modification appears in each position of the genome. And we were doing a lot of basic science of understanding the pathways involved in reading and writing those modifications. And all of this was done in the very nice organism called baker's yeast, which is a wonderful organism. It is well behaved. It doesn't complain. It's easy to manipulate and so on. And about 7 years ago, we made a mistake. And so the mistake relates us to blood. So I'll try to kind of connect us to that mistake. So we're doing a small jump and then we'll return to chromatin in a second. So there is something called cell-free DNA. DNA is the DNA, the genomic sequence, or the molecule. And cell-free means that it is not within a cell. And that word has been used in biology in two senses. One is when we do a reconstruction of systems that behave like a cellular system, but they are in a test tube. We'll put that aside. Or in a physiological setting where they're outside the cells. And when we talk about cell-free DNA, we're usually talking about pieces of DNA that are in the circulation, in the blood, and they get there from cells that died within the body. Okay, so they're not within a cell, they're essentially debris of cells that died. And you know, there's a big mass of how many cells die in our bodies every day. It's perfectly normal to have this turnover of cells. And these cells, some of them end up in the bloodstream, and we can find pieces of their DNA in the bloodstream. So why is that interesting? It's interesting because this is essentially a survey mechanism for abnormal events that happen in the body. And this concept of liquid biopsy that says instead of trying to get physical access to an organ, we'll try to look at debris from that organ in the blood and save the need for physical, essentially invasive surgery, might be useful, came about, is a big thing. There are many companies that make the news now and then about it. Prenatal diagnosis with circulating cell-free DNA is already, because that was not cell-free DNA, it was, we're not going to Tyrannus. So, non-invasive prenatal diagnosis is the diagnosis of essentially the developing fetus that is done from a blood test of the mother. It's actually it's used a lot by now. And there are all those companies that try to do detection and early detection of cancer, recurrence of cancer, and so on, using this idea. Now, both of those applications rely on something very important, is that both the fetus and the tumor, and also the third application, which is transplantation, we're talking about detecting a DNA from an organism that has different sequence than the patient, or the mother is not a patient, is from the host, let's say. So the challenge is what happens when the sequences are the same. We cannot rely on mutations in cancer in order to detect cancer. So we're talking about detecting changes that have to do with, say, liver cell death that has exactly the same sequence as every other cell in our body. So we cannot use mutation anymore. And here comes the idea, and essentially the realization that all this literature on cell-free DNA was showing us pictures like this. But actually, and it's apparently been known since the early day, the picture should be like this. The circulating DNA is not pieces of DNA, it's pieces of chromatin. We can go into the reasons why this is the case, but the idea is that when cells die, they chop up the chromatin and release it as these kind of fragments. And we met these fragments 5 minutes ago. And remember, I told you we were studying it. So the eureka moment was literally when I took this picture. Ronen, who was a scientist in my group, told us about, look, you know, we are hearing talks about cell-free DNA, but it's actually cell-free nucleosomes, and we know how to read nucleosome state. Maybe we can put one together and probe essentially cell-free DNA using the same tools we're using in the lab. And that was in the crazy idea session of a group retreat, so we sat outside instead of in the class. And so the idea is that the modification of the nucleosome, remember I told you, tell us about the activity of that part of DNA in that particular cell. It might differ between cells. The sequence of the DNA is essentially it's an address. It tells where in the genome this came from. So we can relate it back to genes and things like that. And so the idea is very simple. Think again of our very conceptually simple story we had about this albumin gene, which is packaged differently with the green flag for go in the liver cell and the red flag in all other cells. So when these type of cells die, they chop up the DNA into small components. Everything gets mixed up in the circulation, and we collect, so we get a sample of that mixing circulation. And now we're using a trick for enriching only the modification, only the fragments that carry the modification we care about, using a physical reagent that recognize the green flag and not the other flags. Okay, this is now a physical, actual enrichment step. And so we are left only with these populations that are marked with the green flag. And now we take this enriched population, we extract the DNA, sequence it, compare it to the genome, and if we find fragments that map to the albumin gene, we can conclude liver cells have died. Okay? Oh, where's my animation? It's gone. Okay. Lost my animation. Okay, but you're correct. So this is a way of essentially learning about what happened in the body. Okay, so is this so easy? Why nobody did it before? It's not because we're much smarter than everybody else. It's because we came across this idea at the right time, where we and the general scientific community had the tools to work with a small amount of DNA that exists in the blood. Just to list, you know, when you go to the moon, there's immediate reasons why this is impossible. And we were faced with the same thing. You know, the amount of DNA we're talking about is minute, much smaller than everything else that goes around the blood. There are tons of antibodies in the blood, and we were using antibodies to actually catch those modifications. So there's a long list of reasons why it shouldn't work. But as with the moon mission, it's just a question of technology. And we were lucky enough to be in a situation where we had the piece of technology to actually do it. It took almost 6 years to publish this paper that was supposed to be just about showing the method, and ended up, you know, we had to discover a lot of things in order to get accepted. But it appeared in Nature Biotechnology. It made the cover, and you remember the little girl we saw in the first slide? She designed the graphics. So as a happy parent, I'm also proud of this paper for another reason. Okay, and in the remaining 2 hours, I will tell you a bit what we can do with it. How much time do we have?
10 minutes, but a lot of time.
Okay, so I'll tell you a bit more, and then we can wing it based on how much time. Essentially, when Moshe kicks me off. So just to remind you, when we do the pull down, we essentially get this kind of picture. Those bumps are essentially places where we saw pieces of DNA that we sequence and we map them to the genome. So everywhere there is such a bump, it means that there were a lot of sequences that map to this location in the genome. And we use an antibody against a mark. Again, this is alphabet soup, so you're not supposed to remember it. But it's a mark of active promoters. And the notations you see on top are notations of genes, and you can see that these bumps are indeed at the beginning of the arrows, which are where genes are starting. And they only occur in genes that are active in the cell at that particular time. Okay, so here is a region where there are a lot of genes that are active in all cells, so we get a very nice pattern. And we can just look at the names of the genes and collect a region around their start and call this the amount of signal we get per gene, and now we can treat it as a quantitative measurement per gene in the genome. And just to get a sense of what we're doing, since we are not looking at a particular gene, we're looking at all the genes in the genome, we can see a very rich picture. Here is an illustration. You're not supposed to be able to read the names of the genes, but you can see that these three locations, we have peaks that occur in all those samples, which are all healthy plasma from healthy people. But all the ones on the right, we barely see anything in those people. When we go to a zoo of different diseases, samples from people with different diseases, things start to light up in these regions, and now we can connect those genes to activity that happens in cell that died within that person. Now, this is a very unwieldy representation. So instead of showing you all those peaks, we're going to essentially replace each one of those boxes by a pixel with a color that is one heat map, essentially, if it's high or low counts, okay? And so the real data we're looking at is thousands of genes that are rows, different samples, and the color code is I lost my color scale, but yellow is high and blue is low. And you can see that the healthy people are quite similar to each other, but many things grow up in different diseases. And on purpose, I'm not going into the names of the diseases. So here is my first exit prompt. So a summary until now. We have an essay. We've been working on it for years, and we made it reproducible, highly optimized. We've been running on more than 10,000 samples of different conditions by now. So we have a large database of many things, many human conditions. We can start to talk about how we can use it to understand what's going on. We created a company because the only way to make an assay that somebody can actually use in the clinic is you have to be able to order it, and you cannot order it from a lab. So we started a company, and that brings along a whole bunch of things. But the nice thing about it is by now the sample collection and processing is highly optimized. And there's an address you send tubes to, you get data 2 weeks later or 1 week later, depending on the status of the queue. And you get these amazing pictures. And now comes the interesting part, because this is all the setup. We have all this data, and now we have to make sense of it and tie it to the biology of the disease. But since I assume that I'll run out of time at this stage, I'll have the acknowledgement slide for many people that were working on this. For my group and many collaborators, mostly different types of disease collaborators. But if I'll have 5 minutes, I'll give you one vignette of the kind of things we can do.
I'll give you 3.
You'll give me 3. Okay. Tough negotiation here. Okay. So a story that we're trying to publish, we're now revising to hopefully address the reviewers' questions. So there is a chance it might actually appear. This is a collaboration with a group at the NIH. There is a disease called small cell lung cancer. It's the less common type of lung cancer. It's often associated with smoking. It's a really nasty disease. The median survival time is, I think, much less than 5 years. It's usually, it responds real well to the first round of treatment, and then the second round, the disease is essentially doesn't respond, and the progression of the disease is really fast. It's a really nasty disease. And what we got from the group at the NIH, they were doing a lot of clinical trials, and they were collecting plasma, but also tissue samples from the tumors. And so we were able to compare things that we were seeing in the plasma to what they were actually seeing in the biopsies. So we can say, here's a patient that the blood looks like that, and a week later the biopsy was collected and looked like that. So we can start matching things. And that's rare because I'll go back to that. This is one of the largest repository of biopsies in this world of small cell, because usually you cannot collect biopsies you can work with. So very briefly, we were lucky that there was a lot of genes that go up in this disease and show up in the cell-free, in the circulation. This is one of the tumors that releases a lot of cell-free DNA. And we could find genes that essentially are unique to the disease, and we can define a score between 0 and 1, which is essentially tumor load of how much tumor is released into the circulation. And it's really high in patients before treatment. Immediately after treatment, it goes down. It doesn't appear in healthy people. You can divide on the median score in naive patients and predict their survival time. And as you can see, survival, this is the probability of survival at a function of months, is terrible in both cases. So it's kind of useful prognostically. But this is not the interesting part. We could do this with other tools as well. The interesting part, I know I have 1 minute, is that like anything else in medicine, doctors are trying to take a disease and break it down to sub-diseases where they can do differential treatment. So instead of one tool fits all, we find one group where one thing would be useful and another group where something else will be useful. And that's how, roughly oversimplifying a lot of things, how medicine, at least oncology, develops. And in many cancers, this subdivision is based on genetic events in the cancer. And you probably know about personalized cancer treatments. These are essentially looking at genetic mutation in the cancer and finding a drug that attacks that mutation. In this disease, no such connections were found. However, people found subtypes that have to do with the state of the cell, which transcription factor, again, if you don't know what it is, don't worry, drive the state of essentially the progression of the disease. And these subtypes are potentially linked to response to treatment. It's really hard to show that because in order to do the subtype, you have to do molecular assay on the biopsy, and due to the nature of the disease, it's really hard to collect a biopsy you can work with to do those things. So there's kind of a frustrating cycle that you cannot measure something, so you cannot use it in order to try things and so forth. And so it's rarely done in practice. There's very few studies that actually went to the effort. And cutting to the end of my talk, we show that using our plasma sample, we can find genes that are markers in this is in the plasma, that are markers of the different subtypes. And we can show, again, very colorful graph, we won't go into it, that we can classify those even in samples with a small tumor load. And so we are able to give the doctors assessment of the subtype of the patient in a non-invasive way, which is important because A, the invasive way doesn't work, but also during the recurrence of the disease, you will not do another biopsy because that other biopsy, the second biopsy, will actually harm the patient and will not be useful, which is, again, one of the reasons we don't have biopsies is because we usually don't do them. And so this gives us a window to start actually starting trials with the NIH about using these measurements as one of the factors in the decision tree in clinical trials to see if different choices actually would help. Again, this is very early days. I haven't solved the cancer, but it's a positive step towards something useful. And I guess I'll stop now because that's the end of this vignette.